Good evening. Yesterday we started cytology or cell biology, its history and the cell and its parts, and in short I gave the functions of each cell organelle. Now let us study in detail the cell organelles. So today we will start the first cell wall. Cell wall, the Robert Hooke in 1665 first of all observed the cork cells, and it is the outer cover of the plasma membrane. It is a protective cover of plasma membrane. Regarding the chemical composition, the cell wall varies in different kingdoms. In bacteria, the cell wall is composed of peptidoglycan. In fungi, it is chitin. In most of the plants, cell wall is made up of cellulose. Besides these things, beside the cellulose, other chemicals such as hemicellulose, pectin, lignin, cutin, suberin, silica may also seen deposited on the walls of certain plants. Regarding the formation of the cell wall, how cell wall is formed? So during cell division, a phragmoplast is formed between the two daughter cells exactly at the equatorial plate. That form into the cell plate. Now the middle lamella is laid down by the daughter protoplasts on both sides of the cell plate. So first a phragmoplast is formed and later is developed into a middle lamella. Then look here, a cleavage furrow or whatever it may be, whether it is a cleavage furrow or it is a division, at the middle of the cell, daughter cells, a phragmoplast is formed, and it will become the cell plate later, and it divides two cells. Next, regarding the structure, a typical plant cell wall has the following three parts. One is middle lamella, that is the center of two cells, and primary wall, that is this forms below the middle lamella, and then the secondary wall below the primary wall. It forms below the primary wall. So now let us see the middle lamella. As I told you already, it is a thin, amorphous, cement-like layer between two adjacent cells. First layer is deposited during cytokinesis. It is made up of calcium and magnesium peptides besides proteins. So the middle lamella, the chemical composition of middle lamella is the calcium and magnesium peptides and extra proteins also there. Regarding the primary wall, it is found only in meristematic and parenchyma cells. You don't find in remaining cells. First formed cell wall produces the inner to the middle lamella. Inner to middle lamella, the primary wall is formed. In young cells, it is thin, elastic, extensible and capable of growth, which gradually diminishes as the cell matures. Secondary wall is formed before the inner side of this wall. It grows by addition of more wall materials. That addition of wall materials from the existing one, that is called intussusception. So existing one, the wall is formed. How this wall is formed? Now let us see. Here you can find this is the middle lamella. Below the middle lamella, this is the primary wall and then plasma membrane. And a number of microfibrils, cellulose microfibrils are oriented and form the primary wall. Primary wall consists of a loose network of cellulose microfibrils embedded in a gel-like matrix or ground substance. This matrix mainly composed of water, hemicellulose, pectin and glycoproteins. Pectin is the filling material, hemicellulose binds the microfibrils with the matrix, and glycoproteins control the orientation of these microfibrils. Next, secondary wall consists of two continuous interpenetrating systems, the closely arranged systems. One is called cellulose microfibrils and continuous system of microcapillary spaces which may be filled with lignin, cutin, suberin, hemicellulose and other organic substances. So mainly the secondary wall is made up of lignin, cutin and pectin. The secondary wall, it is laid inner to the primary wall after maturity. It is laid down in succession of at least 3 layers. There are many layers, minimum 3 layers. Those are called secondary wall 1, secondary wall 2 and secondary wall 3, of which the secondary wall S2 is thickest one among the 3. So here the secondary wall, it grows in thickness by deposition of materials over the existing structure, that's called accretion. So here you look here, the middle lamella here, middle lamella in the center, middle lamella. Below the middle lamella you have a secondary wall. Below the secondary wall you have 3 walls, S1, S2 and S3 inside the primary, this primary wall, of which the S2 is more thick or thickest among the 3. So this is the middle lamella which separates 2 cells. Inner to the middle lamella, inside the cell, primary wall and then the secondary wall. So while preparation or this is formation of the secondary wall, you can find some thickenings are found in the cells. Formation of secondary wall, etc. In tracheids and vessels, they show various types of thickenings. They may be annular or ring-like and spiral thickenings or scalariform thickenings or reticulate thickenings, and finally the pitted thickenings. Pitted thickenings are found. This is starting from annular to pitted thickenings in an evolutionary way. As a highly developed thickenings are called the pitted thickenings. The most primitive annular thickenings that you can find in tracheids and vessels. Next, plasmodesmata. These are the connections between 2 cells, 2 adjacent cells. The cell wall is not totally complete around the cell. It is interrupted by narrow pores carrying fine strands of cytoplasm which is interlink the contents of the cell. They are called the plasmodesmata. They form a protoplasmic continuum called symplast. Protoplast is called symplast, and it consists of a canal lined by plasma membrane. Now you can see the plasmodesmata. So here you can find this is a cell, this is another cell. So between the cells there are gaps are there in the cell wall. These are called the plasmodesmata, so that there is a continuation of the cytoplasm from one cell to another cell. Plasmodesmata serves as a passage for many substances to pass through. It is also believed that they have a role in the relay of stimuli relay. That's pass the stimulus from one cell to another cell. So the next point is the next side heading, that is the pits. Perhaps you might have known how the pits are formed. See, pits are the areas on the cell wall on which the secondary wall is not laid down. So the left out portions of secondary wall are called the pits. The pits of adjacent cells are opposite to each other. Each pit has a pit chamber, a pit membrane and pit aperture. The pit membrane consists of middle lamella and primary wall. The pit membrane has many minute pores and thus they are permeable. Pits are of 2 types, simple pits and bordered pits. Simple pits, width of the pit chamber is uniform. There is no secondary wall in the simple pit. And having the simple pit is the characteristic feature of the fibers. Fibers having simple pits. The second one is the bordered pit. The secondary wall is partly this is overhangs the pit, that is border the pit, that is the bordered pit. Bordered pits are found in tracheids and vessels. Now you see the center one, it is a simple pit. There you can find central, middle lamella and primary wall. That's all. no secondary wall formation. Secondary wall formation is up to above the pit only. So now the middle lamella and the primary wall is known as the pit membrane. This is called the pit membrane. Pit membrane. And then the space is called the pit cavity. And the opening is called the pit aperture. Opening is called the pit aperture. And whenever there is a pit, you can find an opposite pit. That is called the pit pairs. But when the cell is entered into an intercellular space, there is no opposite pit. Such pits are called the blind pits. Such pits are called the blind pits. And the second one, this is called the bordered pit, where you can find the secondary wall is also arches. This one, secondary wall. And here you can find this is the pit membrane and this is the pit aperture. And this one, pit aperture, is very small when compared to this. Pit aperture is small. And inside you can have pit chamber. So this we have generally while showing here you can find the simple pit. I told you that they are found in fibers. In fiber, if it is a fiber, in a surface view, they are like this. These are simple pits. A small opening, a small hole like this. When it is a bordered pit, here this is a small opening. In a surface view, if you happen to see, these pits are like this. These are called the bordered pits. Bordered pits in surface you can see. In tracheids and vessels, the bordered pit is like this and the simple pit is like this. So these are different types of. Okay. So that you can understand, this is a blind pit, as I told you already. When it enters into an intercellular space, there is no opposite cell, so you can find the pit is only one side. This is called blind pit or off pit or blind pit. These are pit pairs you can find. So bordered pits help in the translocation of substances between the two adjacent cells. Generally, each pit has a complementary pit lying exactly opposite to it in the wall of the neighboring cell. Such pits form a morphological and functional unit called the pit pair. These are called the pit pairs. Next, regarding the functions of the cell wall. So, we know in a plant cell, a definite shape to the cell. This cell wall gives a definite shape. And a protection of a protoplast from injury and infection. Rigidity to the cell. Prevention of cell bursting due to endosmosis. And specialization of tracheids and sieve tubes for the long distance transport. The tracheids and sieve tubes help in the long distance transport. And entry and exit of certain molecules through the semi-permeability. Semi-permeability having the entry of certain molecules through this one. Next to the cell wall, very important, cell membrane, that is exactly below the cell wall. And this cell membrane is found both in prokaryotes and eukaryotes and plant cells and animal cells. So it is selectively permeable since it allows only certain substances to enter or leave the cell through it. So in addition to the eukaryotic cells possess this intracellular membranous collective called the cytoplasmic membrane system. So they are having a number of cell organelles are there double membrane structure, and it is called a total cytoplasmic membrane system. So the membrane of Golgi bodies, endoplasmic reticulum or mitochondria are similar to the structure of the cell membrane. So this is called a cytoplasmic membrane system. So plasma membrane and the subcellular membranes are together known as biological membranes or biological membranes. The membranes of different cell organelles and this cell membrane called biological cell membranes. So now let us see the ultrastructure of this one. The cell membranes are about 75 angstrom units thick under the electron microscope, they appear to consist of 3 layers. An outer electron dense layer, that is 20 angstrom units, and inner electron dense layer is about 20 angstrom units, and a middle pale colored layer, that is about 35 angstrom units. Total, the thickness is 75 angstrom units. The outer and inner layers are formed of protein molecules. So remember, this outer layer, I told you 3 layers. Regarding the 3, outer and inner layers are formed of proteins, whereas the middle one is composed of 2 layers of phospholipid molecules. Phospholipid molecules. So in the center, 2 layers of phosphate, and above you can have a protein layer, below you can have another protein layer. Middle phospholipid layer. Whereas the middle one is composed of 2 layers of phospholipid molecules. Such a trilamellar structure is called a unit membrane, which is a basic concept of all membranes. This is remember, trilaminar structure. So here you see a trilaminar structure of plasma membrane. You can find the outer protein, inner protein, and in middle phospholipid layer. This is the phospholipid layer, and outer and inner, this is protein layer. Biochemical studies on the cell membrane in human red blood corpuscles brought out the detailed structure of plasma membrane. Studies in 1950s revealed that cell membrane is composed of lipids that are arranged in a bilayer. Also, lipids are arranged within the membrane with a polar hydrophilic heads towards outside and hydrophobic non-polar tails towards the inner side. See, here you can find, here you can find hydrophobic. This hydrophobic tail here and hydrophilic head outside. Later, biochemical investigations reported that the cell membrane also possesses protein and carbohydrates. Ratio of protein and lipid varies in different cells. In human beings, the RBC has an approximately 52% of protein and 40% of lipids. Membrane proteins are classified as integral, intrinsic, and peripheral, extrinsic. The peripheral proteins lie on the surface of the membrane. All integral proteins are partially or totally buried in the membrane. This you have to remember. Next, models of plasma membrane. So different biologists gave different models regarding the plasma membrane, of which we can see number 1 is the Denelli and Davison's proposed a trilamellar model. This is a 3 layers model. According to this model, there is a middle clear bilayered lipid layer and outer and inner dense dark colored protein layer. That you know already. This is the model given by Davison. The next model is the fluid mosaic model. This is more appropriate and all are agree with this model. It was proposed by Singer and Nicholson in 1972. According to this model, the plasma membrane is semi-fluid in structure. It consists of a bilayered lipid matrix. The proteins are disposed in a kind of mosaic arrangement in the liquid matrix. So, in the previously we learned that there is a bilipid layer in the middle, bilayered lipid layer. On both sides you can find a protein layer. But whereas here, this is different. Consists of a bilayered lipid matrix, that is okay, no problem, and bilayered lipid, but whereas the protein molecules are arranged in a mosaic manner in this fluid of this bilayered lipid layer. The proteins may be peripheral or integral, whatever it may be. They may be inside or outside. Sometimes they may be outside or they may be intermixed. This is the model proposed by Singer and Nicholson. Is more appropriate. Now you can see this model. Here you can find this. This is the liquid mosaic monomer. This is a bilayered lipid layer. You can find this yellow color, and is red color outside. In this red, this phospholipid layer, integral membrane proteins permanently embedded within the lipid layer, and this here the integral protein that is continuously from top to bottom, and some are peripheral layer you can find in the sides. So here also you can find the outer, you can find this lipid layer, and here you can find the proteins are just intermixed with this one. So this is the fluid mosaic structure of plasma membrane. So regarding the functions, it is selectively permeable, it regulates the movement of materials across it, that is the entrance and exit of materials through the plasma membrane. It is active transport of ions also carried out by plasma membrane. One is the selectively permeable, this is due to diffusion, osmosis it undergoes, and regulate the movement of materials, and active transport of ions also carried out by the plasma membrane. One of the most important functions is the transport of molecules across it. Here you can find two types of transport. One is the passive transport, that is called downhill transport, moving down without any difficulty, without any problem, and without any consuming the energy. The movement of the molecules across the membrane without requirement of energy, without the requirement of any energy from higher concentration to lower concentration. So the process of simple diffusion, osmosis along a concentration gradient are the examples for this passive transport. By that is example water movement by passive transport method. The second one is the active transport, that is uphill, going up, so it needs some energy. So it is energy dependent process in which ATP is used to transport molecules from lower to the higher concentration against concentration gradient. That is that's called the transport of potassium and sodium ions by pumping method. The transporting nutrients into and metabolic waste out of the cell. So regulation of intercellular and intracellular transport. The maintenance of the proper ionic composition, that is pH 7.2, and osmotic pressure of the systole. Semi-permeable or selectively permeable ability ensures that essential molecules, glucose, amino acid, and lipids readily enter into the cell. Another thing is endocytosis and exocytosis. These are two technical terms you have to remember. Endocytosis and exocytosis are active processes involving the bulk transport of materials through the membranes, either into the cell, that is called into the cell is endo, and out of the cell, that is exocytosis. Endocytosis occurs by an infolding or extension of the plasma membrane to form a vesicle or a vacuole, etc. It is of two types. Phagocytosis, that is cell eating, substances are taken up in a solid form. Cells involving in this process are called phagocytes and said to be phagocytes, some which blood cells. And a phagocytic vacuole is formed during this uptake. The second one is pinocytosis, cell drinking. Substances are taken up in liquid form. Vesicles, which are very small, are formed during this intake. So pinocytosis is often associated with amoeboid protozoans and in certain kidney cells involved in the fluid exchange. It can also occur in some plant cells. So here the photograph is given regarding the endo and exo. So the cell wall, this plasma membrane is involved inside, and taking the material and leaving inside. In the same way, taking the waste material and sending outside. That is exocytosis, and this is the endocytosis. Exocytosis is the reverse of endocytosis, by which materials are removed from the cells, such as undigested remains from the food vacuoles. Okay, so now let us go to the next cell organelle, that is cytoplasm. See, it is a gel-like ground substance enclosed within the cell membrane. So first you have the cell wall. Below the cell wall you have the plasma membrane. Below the plasma membrane you will find the cytoplasm only. It is colorless and contain about 80% of water. In prokaryotic cells, all contents are contained in cytoplasm, whereas in eukaryotes, the contents of the nucleus separates from the cytoplasm. Nucleus, because it is having a nucleoplasm inside the nuclear membrane, outside have the this cytoplasm. Whereas in prokaryotes, since there is no nuclear membrane, they are mixed with the cytoplasm. Whereas in these eukaryotes, the contents of nucleus separated from the cytoplasm. So most cellular activities, glycolysis, cell division, occur in cytoplasm. Endoplasm, that is inner granular mass where you can have a cell organelles, and ectoplasm, outer clear glassy layer, outside thin layer below this one without any cell organelles, that is called ectoplasm. And there is a central vacuole you can find. So it has three major elements inside the cell. The first one is the cytosol, that is without organelles. The remaining material is called cytosol, contains 70% of cell volume. The portion contains the membrane bound organelles, not contained. This portion not contained the membrane bound organelles, complex mixture of cytoskeleton, filaments, dissolved molecules, water, soluble proteins. So these are all found in the cytosol. The next organelles usually membrane bound, which are specific functions. So there are many cell organelles and some are membrane bound, which have specific functions in the cell. The third one is the cytoplasmic inclusions, small particles of insoluble substances suspended in the cytosol, ranging from calcium oxalate, silicon dioxide crystals to granules of starch, glycogen, lipids and proteins and other substances, which are all found in the cytoplasm. So cytoplasm also contain within the cytoplasm. This contained within cytoplasm. It is present in all cells, prokaryotic and eukaryotic cells. The cell contents are found. It's a jelly-like material made up of proteins. An elaborate network of filamentous proteinaceous structures are found in the cytoplasm. Those are called microtubules, that I will tell you later. So it is made up of three cytoskeletal filaments. Those are microfilaments, intermediate filaments and microtubules, which are found just like a network in the cell. Network in the cell, that is below this plasma membrane. These are microfilaments, intermediate filaments and microtubules. Functions of the cytoskeleton: because of these things, it forms a structure such as flagella and cilia. It provides mechanical support. It plays an important role in both intracellular transport movement of vesicles and organelles and cell division. That's an important part of this cytoskeleton, these microtubules, etc. What is the function? Regarding this function of cytoskeletons, it maintains the structure of animal cells and shape. It prevents cell from collapses. It prevents the cell from collapsing. Cytoskeletal elements that is interact extensively and intimately with the cell membranes. Synthesis of cell wall in plants helps in movement of organisms, motility in animals. Okay, that's all. By this we finished today the cell wall and plasma membrane and cytoplasm. Today we will deal the remaining cell organelles. Thank you.